Roughly 538 million years ago, pale light filtered through shallow seas, revealing armored bodies moving across a once quiet seafloor. For billions of years, most life remained microscopic. Then eyes, claws, shells, and active hunters appeared within a geological heartbeat. Why did complex animal life diversify so dramatically after an age when Earth seemed almost permanently simple and quiet? The answer lies in oxygen, genes, moving bodies, and a predator-driven arms race that transformed every survival strategy. Hidden among these alien animals, one small chordate-like swimmer carried a structural blueprint that would eventually lead toward us. This was not an empty ocean suddenly filled by magic, but a crowded stage prepared through immense stretches of unseen evolution. Soft sediment covered the seabed, while sponges, microbial mats, and unfamiliar creatures competed for space, food, and protection. The water looked calm. Beneath it, natural selection was accelerating through every bite, escape, burrow, shell, and sensory advantage. Some animals crawled across mud, others drifted above it, and several began hunting bodies that could finally fight back. Fossil evidence suggests this diversification unfolded across millions of years, brief geologically, yet far longer than any human civilization. Its creatures were not failed monsters. They were living solutions to problems that every animal still faces today. Find food or starve, detect danger, escape predators, reproduce successfully, and protect a vulnerable body in an increasingly hostile ecosystem. Each innovation changed the rules, forcing neighboring species to adapt, retreat, specialize, or disappear from the evolutionary contest. The Cambrian seas became laboratories where anatomy, behavior, and ecology were tested at a scale Earth had never witnessed. To understand that transformation, we must return to the long silence before animals began reshaping the planet. Earth formed roughly 4.5 billion years ago, but its earliest environments were violent, unstable, and hostile to complex organisms. Once oceans persisted, simple cells exploited chemical energy, sunlight, and mineral-rich waters, beginning a biological story almost without visible movement. For most of planetary history, life consisted mainly of microbes whose tiny bodies left only subtle chemical and fossil traces. Simplicity ruled everything. Yet microbial communities gradually altered the atmosphere, oceans, sediments, and possibilities available to later organisms. Cyanobacteria released oxygen through photosynthesis, although much of that oxygen first reacted with dissolved iron and exposed minerals. Ancient banded iron formations preserve evidence of this transformation, recording intervals when oxygen changed ocean chemistry on a planetary scale. Complex cells eventually appeared with internal structures, flexible membranes, and genetic systems capable of supporting larger, more specialized bodies. Some cells formed colonies, sharing resources and dividing tasks, while others swallowed neighbors and built powerful internal partnerships. Multicellularity evolved more than once because cooperation offered size, efficiency, protection, and new ways to exploit surrounding environments. Yet becoming multicellular created danger. Cells had to coordinate growth, prevent cheating, transport nutrients, and reproduce without destroying their collective. Early ecosystems still lacked the rapid pursuit, crushing jaws, elaborate eyes, and defensive armor that later defined animal competition. Survival depended on chemistry, sunlight, attachment, slow movement, and access to surfaces where nutrients accumulated. The world was alive, but most of its drama occurred at scales invisible to human eyes. Those quiet billions were essential because evolution was assembling cellular machinery long before fossils revealed its most spectacular results. The Cambrian Explosion began from deep preparation, not emptiness, and every later animal inherited that ancient microbial foundation. Before the Cambrian, Ediacaran seas hosted soft-bodied organisms unlike most creatures living in modern oceans. Some resembled quilted discs, fronds, or flattened mattresses, and many lacked obvious mouths, limbs, heads, or digestive systems. Dickinsonia moved across microbial mats and left feeding traces, suggesting behavior more animal-like than its simple outline first implies. Other forms stood anchored above the seabed, absorbing nutrients or filtering water while currents moved around their delicate bodies. They occupied a quieter ecosystem where active predators may have been rare, limited, or absent across many habitats. Peace had an expiration. As mobile animals spread, stationary and defenseless organisms faced unfamiliar pressure from grazing, disturbance, and possible predation. Trace fossils record increasingly complex movement through sediment, revealing creatures whose soft bodies were rarely preserved. Burrowing mixed oxygenated water into deeper layers, changing microbial communities and destroying stable mats that covered ancient seafloors. This ecological engineering transformed the environment before many engineers themselves appeared clearly in the fossil record. Some ediacaran organisms may represent early animals, while others could belong to extinct branches with no close modern relatives. Their identities remain debated because flattened impressions preserve shapes more readily than muscles, organs, tissues, or genetic relationships. One fossil can support several reconstructions, and each interpretation changes how scientists understand the transition into Cambrian ecosystems. The stakes were enormous. Mobile feeding could unlock new resources, yet movement also exposed animals to detection and attack. By roughly 541 million years ago, many ediacaran communities declined as burrowing, grazing, and environmental change reorganized marine habitats. Their disappearance marks no simple replacement, but a turbulent transition between two fundamentally different biological worlds. Complex animals require energy, and oxygen allows cells to extract far more energy from food than many anaerobic pathways. Geochemical evidence suggests oxygen levels rose unevenly before and during the Cambrian, creating expanding zones where active bodies could thrive. The increase was probably not a single global switch, because shallow seas, deep basins, and coastal waters differed dramatically. Oxygen opened doors. It supported stronger muscles, faster movement, larger bodies, active digestion, and increasingly demanding nervous systems. Changing ocean chemistry also made calcium carbonate and phosphate more available for shells, spines, teeth, and supportive structures. Skeletons offered protection and leverage, but building them required energy, minerals, and biological control over crystal growth. Continental erosion delivered nutrients into shallow seas, potentially increasing primary production and feeding larger, more complicated food webs. Tectonic activity reshaped coastlines, producing broad continental shelves where sunlight, nutrients, and habitat diversity overlapped. Shallow water became evolutionary real estate, filled with gradients of depth, current, oxygen, sediment, and food. Climate shifts may have disturbed ecosystems repeatedly, eliminating established communities while opening space for adaptable newcomers. Snowball Earth episodes had ended before the Cambrian, and their aftermath may have influenced nutrient cycles in ocean circulation. No single trigger explains everything, because oxygen, minerals, geography, climate, and ecology interacted across different timescales. The survival stake was immediate. Animals needing more energy could flourish only where environmental chemistry supported their costly lifestyles. Conditions became permissive, but opportunity alone could not create eyes, limbs, guts, or coordinated behavior. For that, evolution needed a toolkit capable of constructing complex bodies from one fertilized cell. Every animal begins as a single cell, then builds tissues and organs by switching genes on and off in precise patterns. Regulatory genes control where body regions form, helping establish fronts, backs, tops, bottoms, segments, limbs, and specialized structures. Hox genes are especially important because they help organize body regions along an animal's head-to-tail axis. The toolkit came first. Many developmental genes likely originated before their most dramatic anatomical effects appeared in Cambrian fossils. Gene duplication created spare copies that evolution could modify without destroying the original biological function. Small regulatory changes could alter when, where, or how strongly a structure developed, producing major differences in adult anatomy. Segmentation offered repeated units that could become walking legs, swimming paddles, feeding limbs, sensory appendages, or protective plates. A flexible developmental system allowed natural selection to experiment rapidly without inventing every structure from nothing. Yet genes do not evolve alone. Environments determine which body plans survive long enough to reproduce. A novel limb mattered only when it improved feeding, movement, defense, mating, or control of a particular habitat. Development also imposed limits, because changing one structure could disrupt several others built through connected genetic pathways. Evolution explores possibilities, but embryos must remain functional at every stage or the experiment ends before birth. Fossils rarely preserve genes, so scientists compare living animals, developmental pathways, molecular clocks, and anatomical patterns. These lines of evidence suggest the roots of animal complexity extend deeper than the visible Cambrian fossil record. Once ecological competition intensified, the ancient toolkit could generate new forms quickly enough to transform entire communities. Movement changed everything because an animal that could relocate was no longer trapped beside one source of food. Muscles converted chemical energy into motion, while nerves coordinated contractions across bodies that were growing larger and more specialized. Early crawlers crossed microbial mats, leaving trails that preserved behavior even when their bodies disappeared completely. Burrowers moved beneath the surface, finding refuge and food while exposing deeper sediment to oxygenated water. The seabed woke up. Layers once stable for generations became mixed, excavated, grazed, and continually rebuilt. Mobility increased opportunity, but it also created pursuit, because hunters could follow prey instead of waiting for accidental contact. Directional movement favored a front end equipped with sensory organs, feeding structures, and concentrated nervous tissue. This trend, called cephalization, helped create recognizable heads where information and action could be coordinated rapidly. Bilateral symmetry also supported efficient forward travel by organizing paired muscles, appendages, and sensory structures around a central axis. Animals moving through water faced drag, so streamlined shapes and coordinated paddling offered measurable survival advantages. Crawlers needed traction against soft sediment, where poorly controlled limbs could sink, slip, or waste precious energy. Each habitat rewarded different solutions, producing swimmers, walkers, diggers, drifters, climbers, and animals combining several strategies. Failure carried consequences. A slower grazer lost food, while a delayed escape could end an entire genetic lineage. Movement did more than transport bodies. It connected habitats, redistributed nutrients, and intensified encounters between unfamiliar species. Once animals could chase and flee, evolution gained a powerful engine that would never stop accelerating. Light-sensitive cells are ancient, but Cambrian animals developed increasingly sophisticated systems for detecting direction, shape, contrast, and movement. Trilobites built compound eyes from mineralized lenses, creating durable visual organs unlike those of most animals today. Some species possessed wide visual fields, while others reduced their eyes after adapting to dim or buried environments. Vision changed the game. A predator could identify prey at distance, and prey could detect danger before physical contact. Better sight rewarded camouflage, speed, defensive posture, group behavior, and the ability to recognize safe terrain. Anomalocaris carried large compound eyes containing thousands of lenses, suggesting strong visual abilities in well-lit Cambrian waters. Fossil evidence from isolated eyes indicates some arthropods processed remarkably detailed visual information for their time. Seeing required more than lenses. Nervous systems had to interpret signals and trigger coordinated movements within fractions of a second. That neural expense demanded energy, making oxygen availability and efficient feeding increasingly important. Eyes also supported navigation, mate recognition, habitat selection, and timing within daily cycles of light and darkness. A visible animal faced danger, but blindness could be worse when hunters already possessed directional vision. Natural selection therefore refined both concealment and detection, creating an escalating contest between observation and deception. Some creatures hid within sediment, others evolved reflective surfaces, and many adopted shapes that disrupted their outlines. The survival stake became brutally clear: notice first, react first, or become energy for another body. Once the ocean could watch itself, every movement carried information, and information became a weapon. Trilobites appeared early in the Cambrian and diversified into thousands of species across oceans worldwide. Their segmented exoskeletons combined protection with flexibility, allowing many species to crawl, swim, burrow, or roll defensively. A trilobite body included a head shield, articulated thorax, tail region, and numerous paired limbs beneath the armor. Small but unstoppable. Their basic design proved adaptable enough to persist for more than 250 million years. Some grazed microbial films, others processed sediment, and certain forms may have scavenged or hunted smaller animals. Compound eyes help many species scan broad surroundings while maintaining a low profile close to the seabed. Molting allowed growth, but it created a vulnerable interval when the old shell split and new armor remained soft. Fossil beds sometimes preserved discarded exoskeletons in abundance, complicating estimates of how many living animals occupied one site. Enrollment, the ability to curl into a protected ball, shielded softer limbs and undersides from attack. Spines extended the defensive perimeter, making some trilobites harder to swallow, grasp, or overturn. Different sediments favored different shapes, including broad burrowers, streamlined swimmers, and flattened species adapted to unstable surfaces. Their diversity reveals that Cambrian ecosystems already contained distinct ecological roles, rather than one generalized animal community. Competition remained relentless because every feeding strategy depended on limited space, oxygen, nutrients, and protection. Trilobites survived multiple environmental crises, yet disappeared during the Permian mass extinction roughly 252 million years ago. Their long success began in Cambrian seas, where armor, vision, and versatility formed a remarkably durable evolutionary combination. As predators became more effective, prey increasingly invested energy in structures that made capture difficult or digestion costly. Mineralized shells protected soft tissues and provided rigid surfaces where muscles could generate stronger, more controlled movement. Spines created distance. They interfered with grasping appendages, complicated swallowing, and warned predators that easier meals might exist nearby. Small shelly fossils document an early wave of tubes, plates, cones, and armor fragments across Cambrian sediments. Many belong to organisms known only from scattered parts, making complete reconstructions uncertain and sometimes controversial. Hallucigenia carried rows of mineralized plates, while other animals built conical shells or reinforced feeding structures. Wiwaxia combined overlapping scales with long, defensive spines, creating a body that resembled no exact modern equivalent. Defense demanded resources, and excessive armor could reduce speed, flexibility, growth, or reproductive investment. Predators responded with stronger appendages, improved vision, crushing structures, and behavioral strategies for attacking weak points. The escalation became reciprocal. Better protection favored better attack, which favored stronger protection in the next generation. Molting species faced special risk because armor had to be abandoned repeatedly as their bodies expanded. Burrowing offered another defense, but hidden animals still needed oxygen and food while avoiding hunters that could probe sediment. Camouflage reduced detection, although it failed whenever predators evolved sharper vision or different sensory systems. Survival was expensive. Every shell, spine, An escape response consumed energy that could otherwise support growth or reproduction. The Cambrian arms race transformed passive bodies into engineered fortresses, each carrying evidence of attacks that shaped its ancestry. Anomalocaris was among the largest known Cambrian animals, with some species reaching lengths approaching 1 meter. Its streamlined body carried flexible swimming flaps, a fan-like tail, large eyes, and paired grasping appendages near the head. The circular mouth contained hardened plates, although its exact feeding mechanics remain debated among paleontologists. A predator takes command. Anomalocaris could approach from open water, seize smaller animals, and manipulate prey beneath its body. Earlier reconstructions misidentified separate fossils as different creatures because the mouth, appendages, and body were found apart. Only later did scientists recognize those pieces as components of one extraordinary animal, transforming several mysteries into a single reconstruction. Some damaged trilobite shells were once attributed to Anomalocaris, though experiments question whether its mouth could crush heavy armor efficiently. It may have targeted softer prey, smaller arthropods, or animals captured through precision rather than overwhelming bite force. Large compound eyes suggest visual hunting, especially in shallow environments where sunlight penetrated the water column. Swimming flaps generated waves of motion along the body, allowing maneuverability without the fins familiar from modern fish. Its success depended on coordination, because poorly timed movements could lose prey and waste costly muscular effort. The predator also faced danger from injury, starvation, environmental change, and competition with other active hunters. Anomalocaris was not a supernatural monster. It was a specialized animal shaped by the same evolutionary trade-offs governing predators today. Its anatomy belongs near the arthropod stem lineage, illuminating how modern jointed-limbed animals acquired their defining features. At the top of Cambrian food webs, it embodied the new reality that animals could hunt deliberately across open water. Opabinia possessed 5 eyes, a flexible frontal proboscis, lateral swimming lobes, and a body unlike any living animal. Its fossils come primarily from the Burgess Shale, where fine sediments preserved delicate structures, usually erased by decay. The proboscis ended in a grasping tip that may have collected small food items from the seabed. Strangeness had purpose. Every unusual feature helped Opabinia sense, capture, process, or reach resources within its environment. 5 eyes may have provided a broad field of view, although scientists still debate the quality of its vision. Swimming lobes moved in coordinated waves, carrying the animal above sediment while avoiding obstacles, impossible predators. A backward-facing mouth beneath the head suggests food gathered by the proboscis had to be transferred carefully. Opabinia demonstrates that evolution does not design toward familiarity. It modifies inherited structures under local pressures and developmental constraints. Many Cambrian animals appear alien because their lineages ended, leaving no modern species to make their anatomy seem ordinary. Extinction erased context. Without living relatives, scientists must infer movement and behavior from compressed remains inside ancient rock. Opabinia was once so unexpected that an early scientific presentation reportedly provoked laughter from part of the audience. Continued study instead made the animal important for understanding relationships near the origins of arthropods. Its survival depended on precision, because a specialized feeding system could become useless when prey or sediment conditions changed. Cambrian diversity included successful experiments, temporary specialists, and evolutionary branches that vanished despite once thriving. Opabinia reminds us that modern ecosystems display survivors, not the full range of forms evolution once produced. Hallucigenia was a small worm-like animal with paired walking limbs below and long defensive spines projecting above its back. Early researchers reconstructed it upside down, treating the spines as legs and imagining soft tentacles waving overhead. The mistake was understandable because flattened fossils distorted orientation, obscured heads, and preserved different structures with unequal clarity. Science corrected itself, Better specimens and comparisons gradually revealed which side walked, where the head lay, and how feeding occurred. Hallucigenia probably moved slowly across the seabed, using clawed limbs to grip surfaces, while spines discouraged attack. Microscopic study revealed tiny teeth around its mouth and within the throat, suggesting controlled feeding on soft material. Its claws resemble structures in velvet worms, supporting an evolutionary connection within the broader panarthropod group. That relationship links Hallucigenia to lineages surrounding arthropods, including animals with segmented bodies and specialized appendages. Reconstruction remains challenging because soft tissues collapse, decay, twist, and overlap before sediments harden around them. A missing shadow can erase an organ, while mineral staining can imitate anatomy that never existed. Every interpretation must fit the fossil, related species, biomechanics, sedimentary context, and developmental plausibility. The survival stake for Hallucigenia was modest but constant. Feed efficiently while avoiding predators in exposed seafloor habitats. Its defensive spines demonstrate how even tiny animals participated in the same escalating contest seen across Cambrian ecosystems. The overturned reconstruction became a lesson in humility, showing that confidence must change when stronger evidence appears. Hallucigenia looks bizarre today, yet its corrected anatomy reveals a coherent animal solving ordinary biological problems. Wiwaxia carried a soft underside beneath overlapping mineralized scales, with long defensive spines rising from its back like lances. Its body combined features that resemble mollusks and segmented worms, fueling debate over where it belongs in animal evolution. Rows of small scales form flexible armor, while larger spines protected the animal without locking its body into a rigid shell. Wiwaxia probably grazed the seabed using a feeding apparatus armed with backward-pointing teeth that scraped soft organic material. That structure resembles a molluscan radula, although similarities alone cannot prove a direct relationship with modern mollusks today. Some researchers instead emphasize similarities with annelid-like animals, keeping Wiwaxia near one of paleontology's most disputed evolutionary boundaries. Fossils preserve scales and spines more reliably than internal organs, so crucial evidence about muscles, nerves, and development remains missing. The animal moves slowly across muddy bottoms, balancing defense against the need to reach scattered feeding patches before competitors. Its armor discouraged attack, but every plate added weight and required minerals that could otherwise support growth or reproduction. Predators could still target the unprotected underside, making posture, terrain, and early warning essential parts of daily survival. A sudden shadow forced Wiwaxia to press its armored back upward while anchoring its softer body against the sediment. Fossil concentrations suggest these animals sometimes occupied favorable patches where food, oxygen, and firm surfaces overlapped within shallow seas. Wiwaxia's strange design was neither primitive nor unfinished. It was a complete solution shaped for one Cambrian world. Its unresolved ancestry shows why family trees become difficult when extinct branches preserve mosaics of familiar and unfamiliar traits. The armored grazer leaves a larger lesson. Evolution often succeeds through combinations that later disappear, leaving no exact modern equivalent. Pikaia was a small, slender Cambrian swimmer whose body preserved some of the earliest convincing evidence for the chordate lineage. Repeated V-shaped muscle blocks lined its sides, while a flexible internal support helped transmit force along the body. Pikaia lacked jaws, mineralized bones, and modern paired fins, yet its organized axis supported controlled swimming through open water. It probably moved by sending muscular waves backward, using its narrow body to maneuver above muddy seafloor habitats. Burgess Shale fossils preserve flattened outlines and muscle segments, but delicate internal structures remain difficult to interpret with certainty. Most researchers place Pikaia among early chordates, although its precise relationship to vertebrates remains debated among paleontologists. Haikouichthys from the Chengjiang biota carried a defined head region, repeated muscles, and possible gill structures. These animals were tiny beside many arthropods, and survival depended on agility, efficient breathing, and access to oxygenated water. They ruled nothing. Their importance lies not in dominance, but in carrying a body plan with enormous future potential. A dorsal nerve cord, flexible axial support, and segmented muscles created a framework later modified into countless vertebrate forms. Modern lancelets retain a simple version of that organization, offering clues to how early chordate bodies functioned without bones. Similarity does not make Pikaia our direct ancestor. Both may instead preserve features inherited from deeper common ancestors. Cambrian chordates diversified within seas dominated by arthropods, sponges, worms, and predators already occupying most visible niches. Their survival stake was coordination. Weak muscle timing or damaged sensory systems could turn open water into an inescapable trap. Inside their modest bodies, the chordate blueprint endured, eventually supporting fishes, amphibians, reptiles, birds, mammals, and human beings. The Cambrian fossil record is selective because most soft bodies decayed before sediment could preserve their outlines or internal structures. Hard shells survive more often, making armored animals appear more common than equally abundant creatures built from delicate tissues. Exceptional fossil sites change that imbalance by preserving soft anatomy, digestive tracts, eyes, limbs, and sometimes fine external surfaces. The Burgess Shale in Western Canada preserves a Middle Cambrian community roughly 508 million years old with remarkable detail. Rapid burial by sediment flows likely carried animals into deeper settings where limited scavenging and oxygen slowed destruction. Compression flattened bodies into dark films, preserving outlines while distorting thickness, posture, and the original relationships between organs. Chengjiang fossils in China are older, roughly 518 million years old, and reveal an early burst of complex animal diversity. Some Chengjiang specimens preserve guts, eyes, nervous tissues, and appendages that vanish completely from ordinary fossil deposits elsewhere. Sirius Passet in Greenland records another Cambrian community shaped by colder waters, different sediments, and its own preservation filters. Emu Bay shale fossils from Australia preserve predators and arthropods, including visual organs and feeding traces recorded in mineralized shells. Each deposit captures one habitat and one burial event, never the complete ocean or every species living nearby. Paleontologists expose these fossils grain by grain because one careless movement can erase anatomy preserved for 500 million years. Digital imaging reveals hidden contrasts, but every reconstruction still depends on judgment about shadows, minerals, compression, and missing tissue. Absence can mislead. A creature missing from one formation may have lived elsewhere, decayed differently, or avoided rapid burial. These fossil windows are extraordinary, but they remain windows, not complete censuses of Cambrian life across the planet. The Cambrian Explosion was not a single morning of creation, but a prolonged evolutionary interval spanning tens of millions of years. Its earliest signals appear in trace fossils and small mineralized parts before many complete animal bodies enter the record. Different groups diversified at different times, so the apparent explosion becomes several overlapping biological radiations viewed through incomplete rock. Early Cambrian stages record increasingly complex burrows, showing that behavioral innovation sometimes preceded obvious anatomical preservation in body fossils. Small, shelly fossils then multiply across shallow seas, documenting tubes, plates, cones, and defensive structures from many uncertain organisms. Trilobites appear later than some trace makers, reminding us that recognizable skeletons were not the beginning of animal complexity. Molecular clocks often place major animal lineages deeper in time, although estimates depend strongly on models and calibration points. The rock record therefore compresses preparation, diversification, and preservation into a pattern sharper than evolution probably felt to living animals. Even 20 million years is sudden geologically, yet it contains countless generations for selection to modify bodies, behavior, and ecosystems. Generation time mattered. Small animals reproducing quickly could accumulate useful changes far faster than long-lived large organisms around them. Ecological feedback accelerated the process because each new predator, burrower, grazer, or filter feeder altered selection for every neighbor. Innovation produced opportunity, and opportunity rewarded further innovation, creating a self-reinforcing expansion of niches rather than one isolated mutation. Some lineages radiated spectacularly, others changed little, and many disappeared before leaving descendants in later oceans of Earth. The explosion describes a pattern in fossils and ecosystems, not an instant when life suddenly discovered complexity from nothing. Its real speed came from ancient genetic tools, meeting new environments, new interactions, and millions of generations of relentless testing. The Cambrian did more than add species. It reorganized ecosystems into layered communities connected by feeding, movement, shelter, and competition. Sponges and other filter feeders raised living structures above the seabed, creating surfaces, currents, shadows, and refuge for smaller animals. Burrowers opened sediment to oxygenated water, but their tunnels also destroyed microbial mats that earlier organisms depended upon. Predators removed vulnerable individuals, altered where prey could feed, and rewarded communities containing better senses, armor, speed, or hiding places. Scavengers and sediment feeders recycled organic material, returning nutrients to waters where microbial producers and filter feeders could use them again. Food chains lengthened as energy passed from microbes to grazers, hunters, scavengers, and larger predators occupying open water above. Reef-like communities built by early calcareous organisms concentrated food and shelter, turning flat seafloors into complex, three-dimensional neighborhoods. Those structures protected juveniles and small-bodied species, although confined spaces could also trap animals when larger predators entered. Bioturbation mixed waste, oxygen, and buried nutrients through sediment, increasing productivity in some zones while destabilizing others nearby. Open water offered another frontier, favoring swimmers capable of maintaining depth, reading currents, and finding prey beyond the seabed. Yet Cambrian seas remained chemically uneven, and sudden oxygen loss could empty productive habitats faster than animals could relocate. Storm-driven sediment flows, shifting shorelines, and volcanic disturbances repeatedly buried communities, forcing survivors into new habitats and ecological roles. The Cambrian was not a stable paradise. Extinction pulses erased successful lineages, even while other groups continued diversifying. After each disruption, surviving animals expanded into abandoned niches, rebuilding food webs with different species but familiar ecological functions. By the Cambrian's end, the ocean had acquired an enduring grammar: predators, prey, reefs, burrows, swimmers, and armored grazers. The Cambrian ended, but its ecological inventions persisted, becoming the basic operating system of nearly every later marine community. Modern arthropods still rely on segmented bodies, jointed limbs, compound senses, molting, and armor first assembled deep in Cambrian history. Molting remains dangerous because growth requires abandoning protection, repeating a vulnerability already faced by trilobites and other Cambrian arthropods. Velvet worms preserve soft, segmented bodies and clawed limbs that echo features once carried by Hallucigenia and related stem penarthropods. Compound eyes evolved repeatedly, but Cambrian fossils show that high-resolution visual competition had already transformed predator-prey behavior remarkably early. Crabs, Insects, spiders, and countless relatives inherited no single Cambrian blueprint, yet they retained its modular logic of specialized appendages. Chordates followed another path, organizing muscles and nerves around a flexible axis that later supported vertebrate skeletons and powerful swimming. The modern lancelet keeps that architecture simple, revealing how coordinated movement can work without jaws, paired fins, or mineralized vertebrae. Fishes, reptiles, birds, and mammals emerged much later, but their bodies still developed using ancient regional instructions inherited from early animals. Sponges also survived, proving that evolutionary success doesn't require speed, armor, or centralized brains when filtering water remains effective. Burrowing continues to mix modern seafloors, renewing nutrients and oxygen while disrupting organisms that depend on stable sediment surfaces. The first sediment engineers changed their world unintentionally, creating ecological consequences that still shape coastal habitats and ocean chemistry. Shells, spines, camouflage, and escape behavior remain costly investments, balanced against feeding, growth, reproduction, and environmental uncertainty today. Every living arthropod, chordate, mollusk, or worm represents one surviving branch beside countless alternatives removed by extinction events. The Cambrian's deepest legacy isn't a collection of monsters. It's a world where animals continually reshape one another's possibilities. No single trigger explains the Cambrian radiation. Oxygen, developmental genes, changing oceans, predation, and preservation probably interacted across long intervals. Rising oxygen may have supported active muscles, larger bodies, and sustained movement, but oxygen alone cannot explain every evolutionary pattern. Animal embryos already used ancient genetic toolkits, allowing familiar developmental instructions to generate radically different bodies when regulation changed. Genes provided possibilities, not outcomes. Ecological selection determined which combinations survived, reproduced, and spread successfully through competitive communities. Predation increased the value of vision, speed, armor, and concealment, creating feedback that rewarded both better hunters and better defenses. Burrowing transformed the seabed, opening oxygenated pathways while destroying stable microbial surfaces and creating new opportunities for mobile animals. Mineral-rich seawater made skeleton building easier for some lineages, but shells evolved for several functions beyond defense alone. Changing chemistry also altered fossilization, making biological diversification appear sharper wherever hard parts and exceptional soft tissues entered the record. Continental breakup expanded shallow seas, producing warm shelves, isolated habitats, nutrient gradients, and repeated arenas for evolutionary experimentation. Yet geography alone cannot create eyes, limbs, or nervous systems. Inherited developmental systems had to make those structures possible. The Cambrian Explosion was therefore a convergence, not a miracle. Biological readiness met environmental opportunity in escalating ecological pressure. Some causes remain uncertain because rocks preserve outcomes better than processes, leaving researchers to test competing explanations against incomplete evidence. New fossils can overturn confident reconstructions, while chemical analyses and imaging continue revealing structures invisible to earlier generations. The strongest explanation is plural. No single cause replaces the others, because evolution accelerated through several reinforcing mechanisms. What remains unresolved is not whether life changed dramatically, but how each cause contributed across different regions and lineages. By the Cambrian's close, animal life had become mobile, armored, perceptive, competitive, and connected through food webs of unprecedented complexity. The period did not perfect evolution. It exposed possibilities, and natural selection preserved only those combinations that survived changing environments. Many famous creatures vanished completely, yet their lost experiments clarify how flexible early animal development had already become. Others left descendants whose bodies still carry segmented muscles, jointed limbs, sensory organs, nervous systems, and defensive structures. Extinction removed most branches, turning today's biodiversity into a surviving fragment rather than evolution's complete catalog of possibilities. Fossils preserve another fragment, distorted by burial, chemistry, erosion, discovery, and the unequal durability of different tissues through time. Together, these fragments answer our opening question. Life went wild when ancient genetic capacity met expanding ecological opportunity. Oxygen helped, minerals helped, genes helped, geography helped, but interaction transformed those ingredients into accelerating evolutionary feedback across oceans. Every new eye changed camouflage. Every burrow changed sediment. Every shell changed the tools predators needed to survive. The Cambrian explosion was therefore not a crowded accident, but a planetary transition into ecosystems driven by animal behavior. Our own bodies remain part of that transition, carrying ancient developmental instructions repeatedly modified across more than 500 million years. The world beneath those Cambrian waves was strange, but its deepest rules now operate inside every complex animal community. If this journey changed how you see early life, subscribe to Eon Earth for more cinematic natural history documentaries. Like the film and share it with someone fascinated by fossils, evolution, or the hidden history beneath our planet. Until our next descent into deep time, remember, the strangest worlds are not imaginary, they are buried beneath us.